Hello, my name is Kendall Graber. I'm a Senior Biologics Specialist with the USDA Center for Veterinary Biologics. I've been asked to introduce the principles of good manufacturing as required by the Virus-Serum-Toxin Act and Title IX of the Code of Federal Regulations, and discuss how the USDA manages compliance to these regulations for animal biologics. I'd like to start with a brief description of the Center for Veterinary Biologics and our position within the USDA. Then I'll provide an overview of 9 CFR and describe some of the specifics regarding the standards outlined. I will also provide some details regarding how we manage compliance through actions such as inspections and product release, as well as providing some insight into risk assessment and prevention of extraneous agents. I will conclude with a brief description of harmonization of standards for veterinary biologics in animal health regulations. The U.S. Department of Agriculture is made up of multiple agencies and offices serving the American people across the country and abroad. APHIS, the Animal and Plant Health Inspection Service, was established in 1972, but much of the mission work of protecting animal and plant health has been conducted under the USDA for more than 100 years, including the regulation of veterinary biologics since 1913. APHIS provides leadership in ensuring the health and care of animals and plants. APHIS strives to improve agriculture productivity and competitiveness and contributes to the national economy and public health. Veterinary services focuses on the health of animals. This includes monitoring, surveillance, and response to animal diseases, import and export services for animal and animal products, and One Health initiatives. The Diagnostic and Biologics Unit directly supports the veterinary services mission by providing internationally recognized diagnostic laboratory support and the regulation of veterinary biologic products. The Center for Veterinary Biologics is located in Ames, Iowa, as part of the National Centers for Animal Health, and is responsible for the regulation of veterinary biological products. We are not to be confused with the Center for Veterinary Medicine under the FDA, which regulates veterinary pharmaceuticals. In 1906, Upton Sinclair's novel, The Jungle, exposed health violations and unsanitary practices in the American meatpacking industry, which contributed to the public outcry leading to reforms in the industry. Around that same time, hog cholera was sweeping through the country, causing devastating losses. The first practical preventive measure, injection of anti-hog cholera serum, was successfully tested in 1907 by the Bureau of Animal Industry Field Station near Ames, Iowa. However, not all products were effective. Significant losses from unregulated manufacture and distribution of anti-hog cholera serum demonstrated a need to ensure the safe and effective supply of animal vaccines and other biological products. As a result, Congress implemented the Virus-Serum-Toxin Act of 1913 to ensure safe and effective animal vaccines and other biological products. The Act requires that anyone who prepares, sells, barters, exchanges, or ships veterinary biological products must do so in compliance with the USDA regulations. I need to note that the term serum is used to reference serum for which immunological claims are made and does not include the other uses of serum or serum products. The CVB implements the provisions granted by the Virus-Serum-Toxin Act to ensure that veterinary biologics are not worthless, dangerous, contaminated, or harmful. Rather, they should be pure, safe, potent, and efficacious. Another important facet is these products should be available for use. The regulation of veterinary biological products includes several significant procedures established and enforced by the CVB. These include licensing or registration of veterinary biological products based on safety and efficacy data submitted and reviewed by the CVB, inspection of facilities, equipment, personnel, processes, and documentation, both pre- and post-licensure, testing, both pre- and post-licensure, serial release or market release of individual cereals, compliance actions taken on an establishment, product, or cereal when violations are determined, and monitoring of product after release through pharmacovigilance. In short, these procedures enacted by the CVB through the authority granted in the Virus-Serum-Toxin Act protect the public from product that is worthless, dangerous, contaminated, or harmful. Or, to put a positive spin on it, to ensure the availability of veterinary biological products which are pure, safe, potent, and efficacious. We regulate biological products, which by definition are all viruses, serums, toxins, or analogous products which are intended for use in the treatment of animals, and which act primarily through the immune system or the immune system response. Treatment includes prevention, diagnosis, or management of animal disease by products which act primarily through the direct stimulation, supplementation, enhancement, or modulation of the immune system or immune response. The scope of products is wide, as the definition of veterinary biologic product is based on immune response and treatment. The CVB works cooperatively with the Center for Veterinary Medicine on regulatory jurisdiction for newer technologies and treatments, such as cancer treatments. This allows the manufacturer to have a clear path to registration of these important products and technologies from the start of registration. The scope of our regulation covers the diagnosis, prevention, and control or treatment of over 223 different animal diseases. These are diseases of pets, livestock, wildlife, and exotic animal species. They include zoonotic agents such as brucellosis and rabies. We work with state and federal animal health control programs dealing with emerging agents. And we also have issued license for product for pre-harvest food safety, such as vaccines for Salmonella in poultry and E. coli in beef. Veterinary biological products are regulated with the customer in mind. The most obvious are animal owners and veterinarians. But the tools regulated by CVB also assist in health programs, such as diagnostic test kits and vaccines used in eradication programs. We work with other laboratories, universities, and foreign regulators. And we also work with the biologics manufacturers as partners in animal health. At the end of fiscal year 2020, there were 86 licensees and permittees regulated by the CVB. These establishments ranged from 1 or 2 people operating at one site to large corporations with thousands of people and multiple sites. There were 96 domestic and 28 foreign manufacturing sites, as well as 33 domestic quarantine sites. And there were a total of 1,579 active products. As a result, in fiscal year 2020, the CVB authorized market release of approximately 16,600 cereals, amounting to over 121 billion doses released into the market. Our authority covers a wide spectrum of products. There are over 527 unique products for at least 223 animal diseases. We are responsible for ensuring that appropriate and adequate standards are used. Many of our standards are outcome-based, reviewing methods to ensure products are not worthless, dangerous, contaminated, or harmful. Application of the regulations must be uniform and consistent, as the industry ranges from large international companies to smaller domestic companies. The same outcome is required. All must ensure the products they prepare for the market are pure, safe, potent, and efficacious. We are tasked with assuring that quality biologics are available, and we must assess the risks of each unique application. I should note here that with regards to extraneous agents that may be in a product, species barriers may not be an effective safeguard, as species-to-species spread may occur. To give you an idea of the range of product that we regulate, here is a list of species for which veterinary biologics are currently authorized for use. From companion animals like dogs, cats, and horses, to production animals like cattle, swine, and chickens, to exotic animals like alligators, elephants, raccoons, and primates. CVB supports the animal health industry and the general health of the U.S. herd by providing quality deliverables as part of the regulatory process. Prior to veterinary biological products being registered through CVB, a thorough evaluation of data submitted from the biologics manufacturer is conducted to support the use, safety, and efficacy of a vaccine. This is augmented with critical testing conducted by the CVB laboratory. Once a product is licensed, CVB continues to regulate and monitor the product throughout the product lifestyle. This occurs on a daily basis with granting market authorizations and monitoring pharmacovigilance reports. Compliance inspections are performed at regular intervals and noncompliance is addressed. CVB also provides world-recognized services to support the marketing and international distribution of products made in the U.S. by developing tests and reagents to standardize testing outcomes, export certification of products, inspection of facilities, and annual training of domestic and international manufacturers and international regulators via the Institute for International Cooperation in Animal Biologics. Although this is a busy slide, it provides an overview of the different sections of the Center for Veterinary Biologics and how we interact with the regulated biologics manufacturers. An open flow of information and samples between the policy evaluation and licensing, inspection and compliance, and the testing laboratory, as well as the biologic manufacturers, allows for accurate assessment and testing. The Center for Veterinary Biologics is committed to quality management as a component of our mission and vision. Oversight and input is the responsibility of every employee and is vital to the success of our program. Our goals are to embrace a forward-focused course of self-assessment, refinement, and responsiveness to change, while committing to the efficient use of resources as a responsible public agency. We want to effectively partner with customers and stakeholders to maximize the availability of safe and effective products, furthering food security through improved animal health. Within the U.S., the Federal Food, Drug, and Cosmetic Act, which is specific to veterinary drugs, not biologics, has the requirements for current good manufacturing practices. The Virus-Serum-Toxin Act, which is specific to veterinary biologics, is regulated by 9 CFR. There is a long history of quality manufacturing requirements and regulations within the United States. Not all GMPs are created equal. There are variations from country to country. 9 CFR is a performance-based, process-controlled regulatory system which is recognized worldwide for export. 9 CFR and GMPs have similar quality manufacturing and testing standards, and they share a common goal as both are intended to ensure consistency of production and testing and provide for pure, safe, potent, and effective biologics for consumers. Title IX of the Code of Federal Regulations enforces the law of the Virus-Serum-Toxin Act. The requirements are mandated for veterinary biologics. It is further defined by regulations and standard requirements as well as policy and practice. Our authorities are granted by the Virus-Serum-Toxin Act and federal regulations such as Title IX, Parts 101 through 122 of the Code of Federal Regulations. In addition, we publish on policy and guidance documents such as Veterinary Services Memorandum, on practice, through documents such as veterinary biologics notices, supplemental assay methods, licensing guidelines, and our inspection and compliance manual, which is located on the APHIS website. One of the areas where 9 CFR and GMP appear to vary on paper is quality management. 9 CFR does not specifically define requirements for a quality unit or internal audits. However, product quality is a requirement, and we have found that many regulated manufacturers utilize quality management systems and self-inspection programs as a mechanism to ensure compliance to the requirements of 9 CFR. Also, 9 CFR does not spell out specific validation requirements, although there are in-process control requirements to assure validity of testing. In addition, in-process validation is valuable as finished product testing moves away from in vivo testing and historical assays to other methods to evaluate purity, safety, potency, and efficacy parameters. While quality management and validation are not defined as requirements by the 9 CFR, compliance with the other requirements of the 9 CFR demonstrate that they are in place. There are in fact many parallels between 9 CFR and current GMP. These include requirements for personnel in 9 CFR 114.7, buildings and facilities 9 CFR 108, equipment 9 CFR 109, documentation 9 CFR 116, material management and supply requirements in 9 CFR 113.50, production including in-process controls 9 CFR 104.5, packaging and labeling requirements 9 CFR 112, storage and distribution 9 CFR 114.11, laboratory controls in 9 CFR 113, product quality review and change control in 9 CFR 102.5 C1, and rejection, reprocessing, complaints, recalls, and returns in 9 CFR 105.3. Sections 1 through 199 of 9 CFR cover the Animal and Plant Health Inspection Service. Subchapter E is for viruses, serums, toxins, and analogous products. Organisms and vectors are in sections 101 through 124. Here's a general overview of how these areas are covered by 9 CFR. 101 is definitions, 102 is licenses, 103 experimental production, 104 permits, 105 suspension, revocation, and termination of licenses, 108 facility requirements for licensed establishments, 109 equipment sterilization and pasteurization, 112 packaging and labeling, 113 standard test requirements, 114 production requirements, 115 inspections, 116 records and reports, and 117 is animals at licensed establishments. Compliance is assured by numerous factors. Test results of every batch of veterinary biologic are submitted by the manufacturer for review. Results are compared with check testing conducted by the CVB Policy Evaluation and Licensing Unit, and each batch is approved or rejected by CVB Inspection and Compliance Unit for marketing. Periodic, unannounced, in-depth inspections of licensed facilities are conducted by specialists trained in quality assurance inspections. Products imported from foreign countries must meet all U.S. standards, and foreign facilities are inspected at the same rate as domestic firms. Additionally, facilities are inspected prior to the issuance of an establishment license. The Administrative Inspection Review is an annual review of all active licenses, all active permittees, and records in comparison to records kept at the CVB. The review validates active licensees, active permittees, and records concerning licensed premises, responsible personnel, and production. The components of this review are specific to each licensee and permittee. The use and performance of veterinary biologics in the field are monitored through analysis of adverse event reports received by the Center. Investigations of alleged violations of the Virus-Serum-Toxin Act or regulations are conducted or directed by trained CVB investigators. As stated previously, one of the ways we manage compliance is through on-site inspections. Veterinary Services Memorandum 800.91 defines 14 categories of inspection. Inspectors can inspect the entire premises of the establishment, all buildings, all biological products, all materials and equipment, the methods used, and any record pertaining to production, testing, storage, disposition, sale, or distribution of veterinary biologic products. I'll give you a brief overview of our processes for several of these items that are comparable to GMPs. But before I do, you may notice that documentation isn't one of the categories. This isn't because it's not important. Rather, it's because it's integral to every category. 9 CFR 116, Records and Reports, covers these requirements. 9 CFR 116.1 requires detailed reports of all information necessary for a complete accounting of activities. It requires concurrent record keeping, recording of when actions took place, and sufficient details of the events throughout the entire process. The records must be legible and indelible, must be detailed enough to provide an understanding of what happened, and must be verified. Now on to some of the categories described in VS Memo 900.91 and some of the related 9 CFR references. Please note that this is just a very general overview and is not a complete list of references or procedures. Requirements for personnel are described in 9 CFR 114.7. Personnel working in areas where products are manufactured and tested should receive training and periodic retraining specific to their duties and the products manufactured. On inspection, we observe operations to determine if employees appear to be adequately trained and supervised so as to be competent in good laboratory techniques and preparation of veterinary biologics products. We also review training records for selected individuals. 9 CFR 102.4 states that an inspection shall be made to determine whether the condition, equipment, facilities, and the like, and the methods used to prepare biologic products are in conformity with the requirements in the regulations. On inspection, we will review the facilities and equipment to ensure they match what is recorded on the filed facility documents. We will verify that the condition of the facilities and the equipment are such as to assure consistency and to assure that the products accomplish the purpose for which they are intended. We also ensure other operations where live biological agents are handled, including organisms used in quality control, are appropriate, contained, and separated as required. In the process of product licensure, a firm must provide an outline of production detailing how the product is prepared. We must have an outline of production on file for each licensed biological product. Preparation of a biological product in a licensed establishment shall be in accordance with the outline of production. The outline of production shall be prepared as prescribed in 9 CFR 114.9. The outline of production is the agreement between the firm and the CVB of how the product will be produced. The outline of production contains 6 sections and is further described in VS Memo 800.206. Standardization of the outline ensures consistent placement of key information used in regulating veterinary biologics. Outlines are to be written in a manner that ensures consistent production and testing methods and should have enough detail to ensure consistent manufacture of all cereals. Section 1, Composition, includes microorganisms used, source and date of ascension of each microorganism, and proportions of each strain. Section 2, Cultures, includes methods of identifying each microorganism, virulence and purity of cultures, the range of subcultures and passages, composition of media, size and shape of containers used for growing cultures, and conditions for incubation of each microorganism or group of microorganisms. Section 3, Harvest, describes the technique of harvesting microorganisms or tissues for production purposes. Section 4, Preparation, describes the method of inactivation, attenuation, or detoxification, composition of preservatives, adjuvants, or stabilizers, assembly of units to make a cereal, amount of antigenic material per dose, filling information, and desiccation information. Section 5, Testing, contains methods for performing final product testing, testing validity criteria, and the lot numbers of potency references and expiration dates. Section 6, Post-preparatory steps, includes items such as site of manufacture for each portion of the product, the type and size of final containers, collection, storage, and submission of representative samples, use, dosage, and route of administration, and product expiration date. 9 CFR 102.5 requires that products are prepared as described in the outline of production. On inspection, we observe production procedures in progress for compliance with the outline of production. We verify that records are complete and that each critical step is listed in the outline of production. We view records of production for accountability and identification by tracing serials and production lots from raw materials to filling. We determine if the manufacturer's record-keeping system provides for unique identification, tracking, and recording of each ingredient, and if safeguards are in place to prevent errors in the preparation of the product. If record-keeping deficiencies are found, we determine if they apply only to that cereal or if there are consistent deficiencies for that product, group of products, or all products. We will select cereals and determine if they have been prepared according to the version of the outline of production in effect when the lot was presented to CVB for market release. 9 CFR 112.1 states that only approved labels should be used and must be used correctly. On inspection, we review the methods used to ensure the correct label with the correct serial number and expiration date are used for each labeling and packaging event. We want to ensure that the firm maintains accountability for all labels printed for use on licensed product. Licensees or permittees are prohibited from marketing a cereal of product until they receive notification from APHIS of the disposition authorized the market release or shipment of that cereal. Licensees or permittees are prohibited from marketing a cereal of product until they receive notification from APHIS of the disposition authorizing the market release or shipment of that cereal. 9 CFR 116.2 requires firms to maintain detailed disposition records. On inspection, we will evaluate the firm's inventory control prior to release and during distribution, and will evaluate if the product has been distributed prior to approval for release. We will review distribution records for accuracy and adequacy for inventory control and determine if the firm could carry out a total stop sale or recall down to the user level should it become necessary. We will also determine if cooler space is adequate for licensed product at the normal level production and confirm the storage temperature is maintained within the acceptable temperature limits. VS Memorandum 800.91 also lists a miscellaneous category. Under this category, we review the firm's pharmacovigilance program. We'll review the firm's adverse event reports and evaluate investigations and actions taken. We also ensure that final product samplers are adequately trained and authorized, as well as verifying storage locations and storage conditions of APHIS reserve samples. We review investigation procedures for process deviations and quality effects, including procedures for root cause analysis as described in VS Memorandum 800.210. Then we will review reports to determine if the investigation procedure was followed and review evaluation of effectiveness of the corrective and preventive actions. We will also verify that products found unsatisfactory have been destroyed and properly reported. Determine if there was an investigation as to why the product was found unsatisfactory for marketing, and if any corrective or preventive actions were taken. Another parallel I mentioned earlier are material supply requirements. One of the ways to ensure quality is through production standards such as requirements for starting materials. 9 CFR 113.50 requires all ingredients to meet accepted standards of purity and quality. 9 CFR 113.53 lays out the requirements for ingredients of animal origin. Specifically, items not subjected to heat sterilization or other approved sterilization methods must be tested for extraneous agents by acceptable methods. This testing serves as one of the mechanisms in the control of adventitious agents. The accepted test methods are described in 9 CFR 113. The general testing requirements for animal origin raws are sterility in 9 CFR 113.26, .27, and .28, which show methods for testing for bacteria, fungi, and mycoplasma. General viral testing in 9 CFR 113.46, which describes methods for testing for cytopathic and heme-absorbing agents, and 9 CFR 113.47, which describes fluorescent antibody testing for specific virus depending upon the source species of the material. 9 CFR 113.47 defines specific diseases to be tested for based on the source of the materials. The regulation lays out specific requirements for testing of diseases which may be present in the animals from which the materials were derived. Some examples are bovine, caprine, and ovine cells tested for blue tongue virus, and porcine origin for porcine parvovirus. This provides an additional level of protection based on the risks posed by each species. 9 CFR 113.47 also defines specific diseases to be tested for regardless of the species source of the materials. All animal origin raws shall be tested for bovine viral diarrhea virus, reovirus, and rabies virus. Testing is performed on a cell line of the same species of origin as the ingredient, and any evidence of cytopathic effect results in an unsatisfactory result. Master seed testing is performed on a cell line of the species for which the vaccine is recommended. While some species barriers may exist, infectious agents can and do cross species and pose a risk to susceptible hosts. In addition to testing requirements, VS Memorandum 800.51 defines sourcing requirements for ingredients of animal origin. Specifically, animal origin raw materials are to be sourced from the United States or countries considered free, low risk, or not affected with foreign animal diseases of concern, and with negligible or controlled risk of bovine spongiform encephalopathy according to APHIS animal disease status designations. The animal disease status can be found on the USDA's website. VS Memorandum 800.51 lists 5 foreign animal diseases of specific concern. They are foot and mouth disease, African swine fever, classical swine fever, highly pathogenic avian influenza, and Newcastle disease. No ingredients of mammalian origin may be sourced from a country if foot and mouth disease, African swine fever, or classical swine fever is present in any region of the country. If an outbreak of any of these diseases occurs in countries providing ingredients of animal origin, the CVB must be consulted for an assessment of the risk. A risk assessment, including an analysis of the risk of contamination to licensed biologics, must be submitted to the CVB for review to justify using an ingredient of animal origin from a country that has a foreign animal disease of concern. This guidance does not abrogate any other import requirements. All national import and export services restrictions still apply. In addition to testing of starting materials, the CVB also requires final product testing prior to marketing release. Section 5 of each outline of production must indicate the stages of preparation of the biological product at which the samples are collected. The outline must reference all applicable standard requirements. Again, these are described in 9 CFR 113. The outline must also describe all additional tests in detail and state the minimum requirement for each satisfactory test. And finally, each product must be tested as described in the outline. 9 CFR 116.7 requires detailed records of all tests conducted on each cereal and subcereal to be maintained by the licensee. It also states that summaries of the tests shall be prepared and submitted to the CVB using an APHIS Form 2008 or an acceptable equivalent prior to release of the cereal or subcereal. I should also mention that while on inspection, we review laboratory records for testing performed, especially testing required by the outline of production or regulations. These include selected tests for raw materials and other ingredients, bulk lots, cereals, seeds, cell stocks, and diluents. Licensees or permittees are prohibited from marketing a cereal of licensed product until they receive notification from CVB of the disposition authorizing the market release or shipment of the cereal. A sufficient quantity of samples to conduct potency testing are submitted to the CVB along with the APHIS Form 2008 documenting satisfactory test results. A portion of the samples are stored, allowing us to maintain a repository of all cereals released to the market. Both submissions are required for a cereal to be considered for eligibility for release. Cereals may be tested by the CVB laboratory, but not all cereals are tested by the CVB. The selection may be random or based on risk, and the cereal may be tested for more than one release parameter. If the cereal is selected for testing, the cereal is not eligible for release until the testing is complete, the results are validated, and the data is entered into the CVB database. The APHIS Form 2008 with test information is reviewed for compliance to the outline of production. The disposition of the cereal is evaluated and recorded on the form. The form may be audited back if it's incomplete or if additional information is required for evaluation. When the 2008 is processed, the laboratory test report is sent back to the firm as part of the information. In addition to inspections and review of cereals prior to release, the CVB performs other post-licensing activities. Compliance activities include product surveillance through pharmacovigilance, stability monitoring by testing selected products prior to expiration, and investigations when compliance issues are reported. We also exercise some regulatory flexibility, such as evaluating requests for extension of dating on case-by-case basis, and we evaluate requests to reprocess product which may not have met one of the parameters to the outline of production, as well as requests for changes in production processes or facilities. We recognize that there are countries which require GMP certification prior to import of product. This slide provides examples of what we are able to provide. When requested, we provide documentation that a firm is authorized to manufacture, package, and sell veterinary medicinal products in the United States using the principles of good manufacturing as required by the Virus-Serum-Toxin Act and regulations pursuant to that act. We also provide documentation that specific products are manufactured in accordance with the principles and practices of good manufacturing as required by the Virus-Serum-Toxin Act and regulations and standards pursuant to the Act. We have also recently implemented a certificate of inspection similar to the GMP certificates issued by other regulatory bodies after on-site inspections. When exported to other countries, products from the United States are subjected to many international standards. Some of the more recognizable are the ISO standards and those of the World Organization for Animal Health, the OIE, which issues manuals of standards. The CVB provides comments to the OIE and is engaged in creation of documents. The CVB also serves as a reference laboratory for rabies vaccines. It may be noted that although structured somewhat differently, many of the texts of GMP regulations are similar to the ISO standards. There has been some movement towards international harmonization of quality standards for veterinary biologics. These focus on the international standards for quality, including technical harmonization of processes and recognition of equivalence. Harmonization is a benefit since individual requirements can result in duplicate and redundant testing. The Center for Veterinary Biologics is involved with several groups involved with harmonization, such as the International Cooperation on Harmonization of Technical Requirements for Registration of Veterinary Medicinal Products, the Canada-U.S. Trade Agreement, and the Committee for the Americas for Veterinary Medicines. VICH is the International Corporation on Harmonization of Technical Requirements for Registration of Veterinary Medicinal Products. It was established in 1996 and is under the auspices of the World Organization for Animal Health. It is a trilateral involving the EU, Japan, and the United States, although Australia, New Zealand, Canada, and South Africa also participate. VICH provides a basis for wider international harmonization of registration requirements. The working groups within the VICH include biologics quality, biologics stability, good clinical practice, pharmacovigilance, and target animal safety. Canada and the United States have a history of cooperation and communication due in part to the similarity of their programs. Some of the goals that have been established are mutual recognition of serial release testing and laboratory activities, as well as mutual recognition of inspection and licensing requirements, and a coordinated review of applications and information exchange. Currently, we exchange information based on manufacturer approval, and we post French-Canadian translations of labeling materials on our website. The Committee of the Americas for Veterinary Medicine, CAMAVET, was established in 2000. It establishes representatives from the OIE in the Americas, regulatory entities, and the veterinary industry. It functions to support the international harmonization of registration norms and control of veterinary medicines. We are committed to harmonization and continue participation in international meetings and exchanging reference materials and information. It is critical to maintain a transparent process for risk assessment-based international marketing of biologics. And although we are not stated as GMP, the 9 CFR standards are recognized worldwide. So in conclusion, as I have demonstrated, the 9 CFR principles of good manufacturing result in product quality standards which are comparable and accepted as such by many of the regulatory bodies throughout the world. Additional information on CVB and our regulations can be found on our website at aphis.usda.gov. We are the National Center for Animal Health, striving to meet the needs for animal health research, diagnosis, and product evaluation.